Imagine a microscopic instruction manual hidden within every cell of your body. This manual is not just any ordinary set of instructions. It is a master plan that has been fine-tuned over billions of years of evolution. This manual, called DNA, holds the secrets to your existence. It is a long molecule that contains our unique genetic code, the very essence of what makes us who we are. It dictates your height, the color of your eyes, and even your predisposition to certain diseases. Every trait you inherit, from your physical appearance to your susceptibility to certain conditions, is encoded in this remarkable molecule. DNA is the blueprint of life, a complex molecule that orchestrates the symphony of life from the simplest single-celled organism to the most complex beings in the universe. It is the thread that connects all living things, weaving a tapestry of life that spans the entire planet. For centuries, humanity has pondered the mysteries of inheritance. Philosophers and scientists alike have sought to understand how traits are passed down through generations. How do traits pass from one generation to the next? What mechanisms ensure that children resemble their parents, yet also possess unique characteristics? What makes us unique as individuals? Why do we each have a distinct combination of traits that sets us apart from everyone else? The discovery of DNA in the 20th century revolutionized our understanding of life itself. It was a breakthrough that opened up new frontiers in biology and medicine. It unveiled a world of intricate molecular machinery that governs our very being. The processes of replication, transcription, and translation are all orchestrated by this molecular marvel. DNA's story is one of remarkable elegance and complexity. It is a molecule that not only stores information, but also has the ability to replicate and repair itself. It is a story written in a language of 4 letters, a code that scientists are just beginning to decipher. Each sequence of these letters holds the key to understanding the biological instructions that govern life. Yet the more we learn about DNA, the more we realize how much more there is to discover. Each new finding opens up a multitude of questions, leading us deeper into the mysteries of life. This is the journey we are about to embark on, a journey into the heart of life itself. We will traverse the intricate pathways of DNA, exploring its structure and function. We will explore the structure of DNA, its function, and the profound implications of unlocking its secrets. Understanding DNA has the potential to revolutionize medicine, agriculture, and even our understanding of what it means to be human. We will delve into the ethical dilemmas that arise as we gain the power to read and even rewrite the code of life. The ability to manipulate DNA brings with it profound ethical questions that society must address.
Picture a ladder twisted into a spiral. This is the iconic double helix structure of DNA, an elegant design that belies its profound implications. It's a marvel of nature, a blueprint for life itself. The rungs of this ladder are made up of pairs of chemical bases: adenine, thymine, guanine, and cytosine. These bases are the fundamental building blocks of genetic information. These bases always pair up in a specific way: adenine with thymine and guanine with cytosine. This specific pairing is essential for the accurate transmission of genetic information. This predictable pairing is crucial for DNA replication and the preservation of genetic information. During replication, the DNA molecule unwinds, and each strand serves as a template for the creation of a new complementary strand. The sides of the DNA ladder are formed by a backbone of sugar and phosphate molecules. This backbone is like the scaffolding that holds the entire structure together. This backbone provides structural support and stability to the molecule. Without it, the delicate balance of the DNA structure would be compromised. The entire helix is coiled tightly, allowing vast amounts of genetic information to be packed into the tiny nucleus of a cell. This compact packaging is essential for the efficient storage and retrieval of genetic information. The beauty of DNA structure lies in its simplicity and elegance. Despite its complex function, the design is remarkably straightforward. Just 4 chemical bases arranged in a specific sequence can encode the instructions for building and maintaining an entire organism. This sequence is like a biological code, a set of instructions that guide the development and functioning of living beings. The precise order of these bases is what determines your unique genetic code. This code is what makes you who you are, influencing everything from your physical traits to your susceptibility to certain diseases.
DNA is often called the language of life, This intricate molecule carries the genetic instructions used in the growth, development, functioning, and reproduction of all known living organisms and many viruses. But what exactly is this language and how does it work? How do these sequences of nucleotides translate into the complex structures and functions that sustain life? Within the long strands of DNA lie segments called genes. These genes are like sentences in the language of DNA, each one carrying a specific set of instructions. These genes are the functional units of heredity, carrying the instructions for building proteins. Proteins are essential molecules that perform a vast array of functions within organisms. Proteins are the workhorses of the cell, carrying out a wide variety of tasks. They're involved in virtually every process within cells, from providing structural support to facilitating chemical reactions. They form the structural components of our bodies, catalyze biochemical reactions, and play critical roles in the immune response, and transport molecules within and between cells. Without proteins, cells would not be able to maintain their shape, replicate, or respond to their environment. The specific sequence of bases within a gene determines the amino acid sequence of the protein it encodes. This sequence is crucial because it dictates the protein's structure and function. The process of using DNA to build proteins is a fascinating one. It involves multiple steps and various cellular machinery to ensure accuracy and efficiency. First, the DNA sequence of a gene is transcribed into a messenger RNA molecule. This step is known as transcription and is the first stage of gene expression. This mRNA molecule then travels out of the nucleus and into the cytoplasm, where it serves as a template for protein synthesis, where it is translated into a protein. This final step, called translation, occurs in the ribosome and results in the formation of a functional protein that can perform its designated role in the cell.
In 1990, scientists embarked on an ambitious endeavor to map the entire human genome. The Human Genome Project, completed in 2003, stands as a testament to human ingenuity and collaboration. It revealed the sequence of the 3 billion base pairs that make up our DNA, providing a blueprint of our species. This monumental achievement opened up new frontiers in medicine, genetics, and our understanding of human evolution. It allowed us to identify genes associated with diseases, trace our ancestry through genetic markers, and gain a deeper appreciation for the unity and diversity of life on Earth. The Human Genome Project also spurred the development of powerful new technologies for DNA sequencing. These advances have made it faster, cheaper, and more accessible to analyze DNA, paving the way for personalized medicine, genetic testing, and other groundbreaking applications.
Section 5. Revolutionizing medicine DNA as a healing tool. The ability to read and understand our DNA has ushered in a new era of medicine. Genetic testing is now used to diagnose inherited diseases, predict an individual's risk of developing certain conditions, and tailor treatments to a patient's genetic makeup. Gene therapy, a revolutionary approach to treating genetic diseases, involves replacing faulty genes with healthy ones. While still in its early stages, gene therapy holds immense promise for curing diseases like cystic fibrosis, sickle cell anemia, and Huntington's disease. Pharmacogenomics, the study of how genes affect a person's response to drugs, is another exciting area of research. By analyzing a patient's DNA, doctors can prescribe the most effective medication and dosage, minimizing side effects and improving treatment outcomes.
Section 6. Unraveling Ancestry. DNA as a time machine. Our DNA is a living record of our ancestry, carrying within it the stories of our ancestors and the journeys they took. By analyzing specific genetic markers, scientists can trace human migration patterns over thousands of years, revealing the interconnectedness of our species. Direct-to-consumer DNA testing kits have made it possible for individuals to explore their genetic heritage, connect with distant relatives, and learn about their ancestral origins. These kits analyze a person's DNA for specific markers associated with different geographical regions and populations. While ancestry DNA testing has its limitations, it provides a fascinating glimpse into our past, highlighting the rich tapestry of human history and the migrations that have shaped our world.
Epigenetics, well, it's a field that explores how our genes can be turned on or off by environmental factors without changing the underlying DNA sequence. This means that our lifestyle, diet, and even stress levels can influence our genetic expression. Epigenetic changes can be passed down to future generations, impacting their health and development. Understanding this powerful mechanism opens up new possibilities for personalized medicine and preventive healthcare. By studying epigenetics, scientists hope to develop targeted therapies that can modify gene expression to treat diseases like cancer, diabetes, and mental health disorders.
As we navigate the DNA revolution, we must address the ethical challenges and implications that come with it. The potential for genetic discrimination, privacy concerns, and the misuse of genetic information are significant issues that need careful consideration. It's crucial to establish guidelines and regulations to protect individuals' genetic data and ensure that advancements in DNA technology are used responsibly. Public awareness and education are key to fostering an informed society that can engage in meaningful discussions about the ethical aspects of DNA research. By promoting transparency and accountability, we can harness the benefits of the DNA revolution while safeguarding our rights and freedoms.
Looking ahead, the future of DNA research holds immense promise and responsibility. Advances in genomics have the potential to revolutionize medicine, agriculture, and even environmental conservation. However, with great power comes great responsibility. It's up to us to ensure that the benefits of DNA technology are accessible to all and that ethical considerations guide our progress. By fostering collaboration among scientists, policymakers, and the public, we can build a future where the potential of DNA is realized for the greater good. Together, we can unlock the mysteries of our genetic code and create a world where innovation and ethics go hand in hand, paving the way for a brighter, more equitable future.